this.
So thank you.
You are part of this. Thank you so much. She's part of it. So
Thank you so much.
thank you so much. Don't forget to take it afterwards. Faisal. Come, please. I need your help. Now it's solved. Thank you. Because I had a problem. Now it's solved. Okay, assalamu alaikum everyone. Maybe we should start. It's a very big crowd. So I'm not sure how I'm gonna take questions this way. Wow. Huh? Yeah, but we are limited by time, right? Okay, so let's start. So today's lecture is very nice one. It's about the membrane structure and transport. So this lecture will be divided into 3 parts. The first part, we will talk about the structure of the membrane. Then the second part, we'll talk about the different the different ways things are transported across the membrane. And the third part, the final one, is about the transportation of water across the membrane, okay? So let's talk about the first part, the structure of the membrane. As you guys are already familiar with this, is that the cell is surrounded by a membrane, which serves very important function, that it keeps the cell isolated from the extracellular environment. And so in this way, it will keep the composition of the cell, okay, isolated in order for the cell to sustain life, its function. And so the membrane serves multiple functions. One of the functions that the membrane does is that we know that there are proteins that are expressed on the surface of the membrane, right? So these proteins facilitate, communication between cells. So the membrane enables the cell to communicate with other cells, okay, via these proteins that are expressed on the cell surface. Other functions, including the import and export of materials. This is also facilitated by the membrane via multiple ways, as we will learn during the lecture. The third thing is that the membrane is not rigid, isn't it? It has certain fluidity to it. And so the membrane enables the cell to move as well as to expand during cell division. So the cell membrane is very important and it serves multiple functions. But mainly what we're going to focus on today is the transport of materials across the membrane, okay? So if we look at the structure of the membrane, so here we have a cell, and so this is the plasma membrane. So if we zoom into this, into the membrane, what we will see is this. So basically it consists of 2 polysheets of lipid molecules. So these red molecules that you see here. So this is sheet 1, okay? and this is sheet 2. So it consists of 2 polysheets of lipid molecules, and within the membrane we have embedded proteins, which you can see here in green. Okay, so mostly majorly of the lipid molecules as well as proteins. So these lipid molecules are basically, if we're going to take one of those lipid molecules, they are said to be phospholipids. They are called phospholipids, and they have a hydrophilic head, okay, and a hydrophobic tail. Sorry. And a hydrophobic tail, okay? So hydrophilic meaning that it's, you know, it loves water, basically, and hydrophobic meaning that it hates water, okay? And so the main types of lipids which the membrane consists of is the phospholipids, which consist of what? Anyone? Sorry, what are the phospholipids? Anyone? Come on, you guys. The phospholipids, what do they consist of? Okay, exactly. So a hydrophobic tail and a hydrophilic head. Very good. Okay. And so because of this property, because of this structure, okay, because of this structure, the membrane is organized in a way where, or the lipid molecules are organized in a way where the tails are facing each other, and the heads, because they are hydrophilic, they are facing the aqueous solution, which is both exist in the intracellular as well as the extracellular environment. So you can see here the lipid molecules. This is the hydrophilic head, and this is the tail. Okay, and this is the hydrophilic head, and this is the tail. And so the tails, they are hydrophobic because mostly they consist of fatty acids. They are facing each other, whereas the heads, they are facing the you know, the aqueous solution that exists in the intracellular and the extracellular environment. And so because of this organization, these two polysheets of phospholipids, they don't like edges, because energetically this is unfavorable for them, okay? And so they tend to organize themselves in a way to form a sealed compartment, okay? And so this is believed how this is believed how the cell membrane, the plasma membrane is formed, okay? Because it is energetically favorable for these phospholipids to form a finite enclosed compartment, okay? And so the lipid bilayer, because of the composition of the hydrophobic tails in the middle and the hydrophilic heads that are facing, you know, both sides of the membrane, because of this, the membrane serves as a selective barrier against certain molecules. So certain molecules, okay, or gases can cross the lipid bilayer very easily, okay? This is very important. So you can see here, we have gases like oxygen, carbon dioxide, nitrogen. These are small and uncharged, and they can easily pass the membrane. No need of help of whatsoever. They can just cross, okay? Steroids can also pass the plasma membrane. Why is that? Anybody? Why steroids? Yep. They are non-polar. Very good. They are non-polar, so they are lipid soluble, okay? And so in this way, they can cross the membrane very easily, okay? Other molecules that they are small but polar molecules they have limited permeability, okay? They can cross the membrane, but very limited, like water can cross, but very limited. Urea, ethanol, these molecules, they do cross the membrane, but to a certain extent. Okay, we'll take questions at some point. Okay. Large uncharged polar molecules like glucose, sucrose, they don't cross the membrane, okay? Ions, charged ions like chloride, potassium, sodium, calcium, protons or hydrogen, they cannot cross the membrane, they don't, okay? So only small lipid soluble molecules, they can easily pass the plasma membrane, okay? You have a question? Yep. So it's not, we are not talking about what enters and what exits the membrane or what enters and exits the cell. We are talking here generally about what crosses the membrane because of their certain property, okay? We are not talking about, you know, what urea is or what, you know, this molecule does. You see what I mean? This is general overview of what can cross the membrane, okay? Is this clear? Okay. So now we know that the major component of the plasma membrane is the lipid, the phospholipids, okay? The other major component of the plasma membrane is the protein, okay, proteins. And so you can see them here, they are everywhere and they serve multiple functions as we will see in a second. Other components are cholesterol. Cholesterol is very important as it stabilizes the lipid bilayer, the membrane. Other components are carbohydrates, okay? But the major components are the phospholipids here in red and the proteins here, which are in blue. Some of these proteins, as you can see, they span the membrane. So they exist, you know, they have two domains, a cytosolic domain as well as an extracellular domain, while some proteins, they only have a one domain. Either, you know, they are facing the cytosol or facing the extracellular environment, okay? And there are proteins where there are carbohydrates attached to them, like the glycoproteins. These very important for cell recognition, okay? So this is what the membrane is mostly composed of. And so the membrane proteins, as I said, they are the most, you know, or the major component of the membrane. Some are integral membrane proteins. Integral meaning that they span the lipid bilayer, as you can see here. They have two domains, the cytosolic as well as the extracellular domain. And we have the peripheral membrane proteins, which exists, which has only one domain, either facing the intracellular or the extracellular. These proteins serves, you know, many functions. Some of these proteins, they help or facilitate the transport of the materials that cannot pass the membrane easily. We said that there are certain molecules that can pass, like gases, like oxygen, carbon dioxide can pass easily the lipid bilayer, right? While other things, as we will learn in a second, like, you know, sodium, potassium, charged ions, they don't pass the membrane. So they need the help of proteins to get transported across the membrane. So these proteins, especially the integral membrane proteins, they help in the transport of molecules or charged ions across the membrane. And so this is what we're gonna focus on in this lecture. Other functions of these proteins include things like facilitating enzymatic activity, cell recognition, signal transduction. You know, some of these proteins are attached to the cytoskeleton inside the cell, so they stabilize the cell, intracellular joining, physically attaching two cells together. So they serves multiple functions. But the main function that we will focus on is the role of these proteins in transporting some of the materials that don't cross the membrane easily. Okay, are you with me? Guys, We should distribute coffee free, for free here. We should, okay? Yep. Very good question. I would encourage you to look. Very good question. Okay. So he is asking if the steroids are facing the hydrophilic head, then how can they, get into the membrane? Anyone has an idea why is that? So he is asking the question that the steroids, they are lipid soluble, right? But the membrane consists of the hydrophilic head, and in the middle we have the hydrophobic tail, right? And so it has to pass the hydrophilic head first to go to the membrane, right? So how does it do that? Okay, yep, you have an answer? Sorry? By? Protein gate? No, they can easily pass. Okay, I will encourage you to look. Okay, because we don't have time for this, I have to run through this, okay? So we'll come back at the end of the lecture if we have time. Otherwise, I would very definitely encourage you to check. Now, let's focus on the transport across the membrane of the different materials. Now, we learned from the previous lecture that large molecules enter the cell by exocytosis, or enter the cell by endocytosis and exit the cell by exocytosis, right? So these are the large molecules, okay? They are transported to the cell by forming the vesicles, okay? However, this type of transport is slow, is not fast enough to meet the demand of the cell for transportation of, molecules that are essential to sustain life of the cell, okay? So there are other mechanisms for transport of ions or, some important solutes. So large molecules, again, are transported by vesicles, but this mechanism is not fast enough for ions and some important solutes. So the other ways of transport are basically categorized into three ways, okay? There is the passive transport, okay, including simple diffusion as well as facilitated diffusion, and there is the active transport. The difference between the two is that with passive transportation, the materials are transported from an area of high concentrations to an area of lower concentrations, okay? So they follow a concentration gradient. And so there is no need for consumption of energy, okay? Whereas with the active transport, materials are being transported or ions being transported against their concentration gradient. So for example, here we have high concentration of for example, potassium, and here we have low concentration of potassium. This protein would transport potassium against its concentration gradient. So it will transport potassium from the low, from extracellular environment where there is low concentration of potassium to an area, to the area where there is high potassium concentrations, okay? So this form of transportation is called active transport. Whereas with passive transport, usually, or it is the transport of ions or molecules down their concentration gradients, okay? And so we have two types of the passive transportation. There is the simple diffusion, where materials will, where, for example, certain gases like oxygen, carbon dioxide, we said they do cross the membrane easily. This is by passive. diffusion, okay? While other molecules or ions, they need the help of proteins to enter the cell, and this is called facilitated diffusion. Okay, is this clear? Okay. Now with simple diffusion, as I said, so certain molecules cross the membrane easily, okay, like steroids, gases, okay, and so they follow, basically they move down their concentration gradients from an area of high concentrations to an area of low concentration. And so in this way, the rate or the speed of transport for these molecules are dependent on how big is the difference between the concentrations of this molecule inside to outside. So if the difference in the concentrations in the outside compared to inside is big, then they will move very fast. As you can see that the rate is dependent on how big is the difference of the concentrations of the solute inside to outside. Okay, I have to move. Okay. So this is passive diffusion, okay, simple diffusion. The other subtype of passive diffusion is facilitated diffusion, as the name states. It is facilitated, okay, meaning that certain molecules need the help of proteins in order to get transported down their concentration gradients. For example, like ions, sodium, potassium, calcium, these are charged molecules. So charged molecules, they don't cross the lipid bilayer, so they need the help of proteins to move from the area of high concentration to an area of low concentration. So facilitated diffusion doesn't require energy, but these molecules do need the help of proteins to move, to inside or outside the cell, okay? And so these proteins can be either, a carrier-mediated protein, as we will learn in a second, or an ion channel. So with simple diffusion, we said it's linear, okay, as you can see here, is dependent on the difference of the concentration inside to outside. However, with the carrier-mediated protein, also is the same, but and it's even faster in terms of the rate of transport than the simple diffusion. But as you can see here, at some point, it stabilizes or become plateau, okay, the facilitated diffusion. Anyone knows why is that? Why it would reach, a point where it becomes a plateau? Yes, yep, you are. Homeostasis, what do you mean by homeostasis? Meaning that reached an equilibrium? No. Still moving, they are still moving, but the speed is just not getting faster, okay? Yep. Very good, excellent. Because these, carrier-mediated proteins, there are limited numbers of them on the membrane, and so it has reached the capacity, all of them are being occupied, right? To transport the molecules to the inside, so it doesn't get any faster, okay? And so this is why it has stabilized, or the rate reached a plateau, okay? And so this is especially true. This is especially true for this type of graph you see here with the plateau is typical of the carrier-mediated protein. The ion channels follow a different law. Okay, so one of the type of proteins that facilitate the, is done by facilitated diffusion is the ion channels, okay? These ion channels, firstly, they enable the movement of ions, charged ions, sodium, potassium, calcium, etc., And so these ion channels are selective. So there are ion channels which are more, are exclusively selective for sodium. Okay, and there are ion channels which only allow, for example, potassium. So they are highly selective. Also, these ion channels, they do, have a gate where they open, a response to a specific stimulus, okay? Some of them, very few of them are non-gated, okay? They are always open, but mostly, most of them, they do have a gate, some of which they do have voltage-gated ion channels, meaning that they open when there is a change in the membrane potential. What do we mean by this is that we know that the membrane, so this is inside the cell, so the inside of the cell is more negative than outside of the cell, as we will learn in the coming lectures. And so these proteins, ion channels, if there was any change in the membrane potential, okay, these ion channels would sense this change in the membrane potential, and they would open their gate to allow the entry or exit of the ions, okay? So these are called the voltage-gated ion channels, okay? And there are channels which are ligand-gated ion channels. These are not sensitive to voltage. These are sensitive, or they open when a chemical agent or a certain protein is bound to them. Okay, so this is why they are called ligand-gated. Okay, for example, acetylcholine. When acetylcholine binds to its, when it's bind to their respective ion channel, it would open and it would allow the movement of ions. Okay, a cholinergic receptor. Okay. Okay, and so this is the second types of proteins that they do facilitated diffusion is the carrier proteins. Okay, these are different from the ion channels. The carrier mediated proteins, firstly, they don't have a opening that span the whole protein. They either open to the extracellular, environment, or to the intracellular environment. So when the molecule is bound to them, this usually causes a conformational change, which will, as you can see here, would open and allow the molecule to enter into the cell. So an example of a molecule that is transported this way is glucose, okay? So glucose is transported by glucose transporters, which are a form of carrier-mediated proteins, okay? Any questions so far? Okay, so what we talked about so far is the passive transport. And so the passive transport, as we said, doesn't require energy, right? So molecules would be moving from areas of high concentration to an area of low concentrations. And so if these molecules are passing the lipid, bilayer, this is called simple diffusion. If it is by the help of proteins like ion channels or carrier-mediated proteins, this is called facilitated diffusion. Now, the other type of transport is the active transport. And so active transport requires energy because it will move the molecules against their concentration gradient. And an example of this is the sodium potassium bump, okay? Because the sodium potassium bump, we know that potassium is highly concentrated inside the cell in comparison to outside, while sodium is mainly concentrated outside compared to inside. And so this pump, what it does is basically it transports 3 sodium, as you can see here, to outside. Okay, so from inside where there is low concentrations of sodium to outside where there is high concentrations of sodium. Along with this, it will transport potassium from the area where there is less of the potassium, which is outside, to the area where there is high potassium to the inside. And so this active pump is very important to keep the composition of the inside of the cell, within a, the concentrations of ions inside the cell within a certain, limits that will sustain life. Otherwise, if the cell was only following the passive transport of materials, then the inside of the cell, the composition of the cell would change, right? Okay, and so the active transport can be, can be divided into 2 types of transport. There is the primary, and there is the secondary active transport. The primary transport, the primary active transport is like the sodium potassium pump, which we just, described, where it will directly uses energy, ATP, to move the sodium and potassium against their concentration gradients, okay? And so this is a primary active transport, which is the sodium potassium pump. The secondary active transport take advantage of the gradient made by the active pump to transport materials to the inside or out, molecules to the inside or outside of the cell. So for example, in this case here, this in green is a secondary active transport where this carrier mediated protein would, transport sodium, would transport sodium down its concentration gradient, okay? So you can see here that sodium is being transported from outside to inside. So outside is high of sodium and inside is low. So this protein would transport sodium down its concentration gradient. Along with it, it will transport a molecule, a second molecule against its concentration gradient. Okay, and so if the direction of transport of the molecules is opposite to each other, as in this case, you can see that sodium is going to the inside and the other molecule is going to the outside. This type of transport is called exchange or antipode, okay? While if the direction for both is going to the inside, if the direction is the same, where, in this case, sodium is being transported to the inside down its concentration gradient, the other molecule is going to the inside against its concentration gradient, okay? But both, in this case, they are going in the same direction, right? So this is called a co-transport or a signboard, okay? Is this clear? Sorry? Okay, I'll repeat that. So you know the active transport, right? The primary active transport. You are good with that, right? Okay. So with the primary active transport, you see here the sodium potassium pump. See that it will transport sodium, both sodium and potassium, against their concentration gradients, okay? While with the secondary active transport, it will only transport the second molecule, against its concentration gradient. So in this case, sodium, this is the both, they are secondary active transport. And in this case, sodium is the main driver, okay, where it will be transported down its concentration gradient. Along with it, another molecule will be transported, which is in this case is S, but S will be transported against its concentration gradient, okay? And so if you look at the arrow, both are going to the inside. They are in the same direction, right? So this is called a signboard. While in this example here, there's another example of a secondary active transport. Sodium is the main driver. So it will be going to the inside, down its concentration gradient. Along with it, S will be going to the outside against its concentration gradient. And so if you look at the arrow, the direction is that opposite to each other, right? So this is called, anybody? Antibord, okay? Clear enough? Now, let me give you an example of some of the secondary active and primary active transporters. Okay. So here we have an example. So in the intestine, for example, we have some cells, despite having a low glucose in the extracellular environment. So here is, we have the cell. Okay. And here outside in the extracellular environment, we have low glucose, high sodium, you can see here, high chloride. Okay. Now we said the primary, one of the most popular example of an primary active transport is the sodium potassium pump, right? So it will pump potassium and sodium against their concentration gradient. So you can see that sodium is being transported to the outside where there is high sodium, okay? And potassium is being transported to the inside where there is high potassium, all right? Along with it, a secondary active transport would be activated. And in this case, this is this sodium here, you can see, is the main driver where it will be transported from outside where there is high sodium to the inside where there is less sodium. So sodium is moving down its concentration gradient. Along with it, it's gonna move another molecule, which is in this case is glucose, against its concentration gradient. Because you can see here that we have low glucose outside compared to inside where there is higher glucose. And so despite that, it will move glucose from outside to inside. And in this case, this type of secondary active transport, as you can see that the arrows are both pointing to the inside. Which type is it? Signboard, very good. So it's co-transporter, right? This one is as well. You can see that sodium is being transported down its concentration gradient. The chloride is being transported again down its concentration gradient. Along with this, it's gonna transport potassium against its concentration gradient. And so in this case, it's also called a signboard, right? Here is an example of an exchanger or an antibody. We know that calcium is very important second messenger, right? Which we said in the previous lecture that it's stored in the endoplasmic reticulum. So when there is a signal, usually the calcium will be released from the stores, and this will increase the concentrations of calcium in the cytosol. Outside, we have an even bigger concentration of calcium, okay? And so calcium would be transported. So here we have the sodium potassium pump, which we said the primary active transport, right? Along with this, we have sodium calcium exchangers, which will move sodium down its concentration gradient to the inside. Along with this, it will expel calcium to the outside against its concentration gradient. Okay, and so this is called an exchanger or an antibody. And this is a secondary active transport. Is this clear? Yep. Yes, sorry, one, yeah, exactly. So one is the main driver, because remember here, the secondary active transport doesn't directly consume energy. It takes advantage of the gradient that is made by the primary active transport. The primary active transport will transport sodium to the outside. It will concentrate sodium in the outside. And so this will make a huge concentration gradient for sodium to go to the inside, right? Because we have high concentration gradient outside, we have high concentrations of sodium outside, right? And so sodium wants to go to the inside, right? And so with the secondary active transport, it will take advantage of this gradient. And so it will operate in a way that it will transport sodium down its concentration gradient. Along with it, it will take a molecule to go against its concentration gradient. Is this clear? Okay. So this was on the different, transport of ions, etc., molecules. Now we'll focus on the transport or movement of water, okay? And so the movement of water is given a different name. It's called osmosis, okay? And although we said that the lipid bilayer have some, permeability to water, but it's very limited. And usually water gets to the inside of the cell with the help of a protein called aquaporins, okay? And so any questions on what we described before we go into the movement of water? Yeah? What do you mean? What do you mean? Energy. Okay. So the energy is directly consumed by the primary active pump. So it will consume energy to move sodium and potassium against their concentration gradients. Whereas the secondary active pump, it will take advantage of this. It is dependent on the primary active transport, okay? Okay, so let's move into water. Let's talk about water movement. And so we said water movement is given a different name. It's called osmosis, okay? It's different than diffusion. When we talk about diffusion, we talk about solutes, okay? Whereas with water, it's a solvent, okay? And so you can see here with diffusion, so these are the solutes, okay? They are being put in a solvent, okay? And so these solutes will move or diffuse from area of high concentrations to an area of low concentrations. It's like when if I put an ink in water, right? It will diffuse, right? Firstly, it will become condensed and then it will just, migrate to distribute evenly, okay? And so this is called diffusion. Whereas with osmosis or water movement, it's different. So you can see here, we have two compartments. This compartment has low solute concentrations, right? Whereas here we have high solute concentrations. Here is set to have high water concentrations because there is low solute concentrations. And here we have low water concentrations, okay? And so water will move from areas of low solute concentrations to an area of high solute concentration, as you can see here, in order to reach an equilibrium, okay? And so the driving force for water to move is called the osmotic pressure, which is which basically is made by the number of particles. So what do we mean by that? We mean, like, for example, let's take this as an example. So here we have high concentrations of solutes, right? And in this compartment here, we have low solute concentrations. This means that this area where there is high solute concentration, there is a high osmotic pressure, okay? And this area where there is less solute concentrations, there is low osmotic pressure, okay? And so water will move from area where there is low osmotic pressure to an area where there is high osmotic pressure, okay? So you can see here that water has moved from this compartment to this compartment. And so the force or the power that it will take to push water to its original level, which is in this case, this one here, is called the osmotic pressure and can be expressed in atmospheres unit, okay? So what you need to know or what you need to learn is that molecules, okay? So let's take this as an example. The osmotic pressure is dependent on the number of particles, not the size of the particles. This is very important. So what do we mean by this? We mean, for example, if I have here a 1 molar of glucose, okay? So let's say here we have 1 molar of glucose, okay? And here we have 1 molar of sodium chloride. Okay, so what do you think? Which compartment would have a higher osmotic pressure? Yep. Very good, excellent. So what he was saying is that with this here, this compartment number 2, so let's say this is 1 and this is 2. So number 2 would have higher osmotic pressure because sodium chloride in water will dissociate to sodium and chloride. So 2 osmotically active particles, okay? And so in this case, water will move from compartment 1 to compartment 2. So similarly, for example, for calcium chloride, this will give to a 3 osmotically active particles, okay? And so if I have here 1 molar of sodium chloride and here I have 1 molar of calcium chloride, the calcium chloride would have more osmotically active particles, thus will have higher osmotic pressure, okay? Is this clear? Repeat what? This one here, okay. So water will move, okay, according to the osmotic pressure, okay? The driving force for movement of water is the osmotic pressure, which is created, okay, by the number of particles, not the size of particles, okay? And so, for example, if we have a let's say here we have similarly like 2 tubes with a selective barrier. Here I have 1 molar of sodium chloride, and here I have 1 molar of calcium chloride, okay? So when sodium chloride is in water, it will dissociate to sodium and chloride, right? Here in this case, it will dissociate to 1 calcium and 2 chlorides, right? And so in this case, we would have 2 active particles, and here we will have 3. And so in this case, because we said the osmotic pressure is created by the number of particles, not the size. So here we have higher osmotic pressure, And here we have lower osmotic pressure. So water will move from here to here. Okay, is this clear? Okay, good. Okay, so you can see here, similarly, just what we explained. So this side 1, and here we have side 2. Here we have low solutes. So this side has lower osmotic pressure. This side has higher osmotic pressure. And so water will move from side 1 to side 2. And so there are terminologies to describe this. So if the osmotic pressure is the same in the two sides, this solution is called isomotic solution, okay? Whereas if the the side where there is higher osmotic pressure, like side 2 here, respective to side 1 would be called hyperosmotic, okay? And side 1 would be called hypoosmotic, where there is low osmotic pressure. Okay, is this clear? And so tonicity is different than osmolality, whereas tonicity is the effect of the solution on the cell volume. So what we described previously are all theoretical scenarios. Okay, when we talk about the cells, we talk about tonicity. Okay, so let's take this as an example. Here we have a cell, and we know an average cell has an osmolality of a 300 milliosmol. Okay, and if we put this cell in water, which is a hypoosmotic solution, right? So what do you think will happen? Yep. The cell will swell. Why is that? Sorry? Exactly. So basically here in the pure water, we have low osmotic pressure, right? Because there isn't much solutes in here, right? Whereas inside the cell, we have higher osmotic pressure. And so water will move from outside to inside, and this will make the cell swell, okay? Whereas in this case here, we have the cell having a 300 milliosmol, in terms of osmolality, and outside we have 1,000 milliosmol. So which side, is it the cell or the solution has higher osmotic pressure? The solution has higher osmotic pressure. And so the water inside the cell will move to the outside, and this will make the cell shrink, okay? And so if the solution make the cell swell, this solution is said to be hypotonic solution, okay? And if it makes it shrinking, it will be called hypertonic solution, okay? And if it doesn't do much or doesn't alter the cell volume, it will be called isotonic, okay? So in this example here, the distinction between osmolality and tonicity is much clearer. For example, let's take this as an example in scenario 1, where we have a cell with a 300 milliosmol, and it was put in a sodium chloride solution of 200 milliosmol. Okay, what will happen is that water will move from area of low osmotic pressure to inside the cell where there is higher osmotic pressure, and this will make the cell, swell, right? And so in this case, this solution is called hypotonic solution. Now in scenario 2, we have the cell, which has a 300 milliosmol in terms of osmolality, and we have a solution which has sodium chloride with 300 milliosmol and urea with 100 milliosmol, right? We know that urea can cross the lipid bilayer easily, right? And so in this case, what will happen is that the cell essentially, so this solution, just looking at the osmolality, we know that altogether this solution has 400 milliosmolality, which is a hyperosmotic. And theoretically, this would make the cell be what? What would happen to the cell in this case when it's put in this solution? It will make it shrink, right? Because the outside solution has a higher osmotic pressure than the inside, right? But what will happen is that initially water will go to the to the outside. Okay, so this initially what will happen, but we know that urea is penetrating and it will move from an area of high concentrations to low concentrations. So urea will go to the inside of the cell along with it. So here we have an increase in the osmolality as urea enters. Along with it, water will move to the inside. And so in this case, the solution, although is hyperosmotic, but in terms of the tonicity, it is an isotonic solution because it did not change the volume of the cell. Okay, is this clear? Yeah. Sorry, I cannot hear. Sorry? Again, the difference between hypotonic and what? Hypoosmotic. Okay. So in this case here, it was a hypoosmotic, solution. But remember here we have only sodium chloride, which is not easily permeable to the cell, right? It doesn't cross the plasma membrane easily, right? It does require ion channels. Whereas in this case, we have the urea, okay, which can penetrate the cell plasma membrane. And so because the urea can cross the plasma membrane, it will help in regulating the volume of the cell. So the solution itself, although it is hyperosmotic, but it did not change the volume of the cell. And so in this case, it's said to be isotonic solution. Is this clear? Yep. Let me explain the next slide. Maybe it will make things much clearer. Let me explain the next slide. Just 1 second, because this is the last one. So you can see here, this is how cells regulate their volume. Okay. So what happens in response to a hypotonic solution? So which will make the cell swell, right? A hypotonic solution has a low osmotic pressure, and so water will go to the inside of the cell, right? And this will make the cell swell. In response to this, the cell, what they tend to do is they will move potassium to the outside of the cell, okay? And so this will make the inside has less solutes, and so water will start exiting to the outside, okay? So this is a mechanism that a cell can use to regulate its volume. Another mechanism in response to a hypertonic solution. So when the cell is put to a hypertonic solution, it would shrink, right? Because water will move from the inside to the outside. But in this case, cells they have a way to regulate this by activating the sodium chloride potassium pump, which is a secondary active transport, to transport ions, more ions to inside of the cell. And so this, okay, and so this will increase the solutes, okay, in the inside of the cell to regulate its volume. And so water will attract to the inside along with this, or they will synthesize more proteins inside of the cell to increase the solutes inside of the cell, and this will attract water to the inside. Yep. Any questions? Yep. Okay, what time is it? Oh yeah, we should go. Huh? Yeah, attendance, sorry. Okay. I'll attend you all. Okay. This is what I will do. We are free to go.
For everyone who has a problem with the attendance, make sure to send us on the document عشان we can mark you as present. خلاص؟